Hello, welcome to Biology, the Scientific Study of Life. 32, an introduction to animal diversity. Welcome to your kingdom. Reading the last few chapters, you may have felt a little like a tourist among some rather unfamiliar organisms such as slime moles, whisk ferns, and sac fungi. You probably are more at home with the topic introduced in this chapter, the animal kingdom, which of course includes yourself, but as this figure suggests, an animal diversity extends far beyond humans or even the dogs, cats, birds, and other animals. We humans regularly encounter. Biologists have identified 1.3 million living species of animals, and estimates of the total number of animal species run far higher, from 10 to 20 million to as many as 100 to 200 million. This vast diversity encompasses a spectacular range of morphological variation, from corals to cacarges to crocodiles. In this chapter, we embark on a tour of the animal kingdom. Let's talk about the animals are multicellular heterotrophic eukaryotes with tissues that develop from embryonic layers. Constructing a good definition of an animal is not straightforward, as there are exceptions to nearly every criterion for distinguishing animals from other life forms. However, several characteristics of animals, when taken together, sufficiently define the group for our discussion. A nutritional mode. Animals differ from both plants and fungi in their mode of nutrition. Recall that plants are autotrophic eukaryotes, capable fungi capable of generating organic molecules through photosynthesis. Fungi are heterotrophs that grow on or near their food, releasing exoenzymes that digest the food outside their bodies. Unlike plants, animals cannot construct all of their own organic molecules, and so in most cases they ingest them either by eating other living organisms or by eating non-living organic material. But unlike fungi, most animals use enzyme to digest their food only after they have ingested it. Cell structure and specialization. Animals are eukaryotes and like plants and fungi, but unlike most protists. Animals are multicellular in contrast to plants and fungi. However, animals lack the structure supports of cell walls. Instead, animal bodies are held together by structure proteins, the most abundant being collagen. Notice this figure. In addition to collagen, which is found mainly in extracellular matrices, animals have three unique types of intracellular junctions: state junctions, desmosomes, and gap junctions that consist of their structural proteins. Among animal cells are two specialized forms not seen in other multicellular organisms: muscle cells and nerve cells. In most animals, these specialized cells are organized into muscle tissue and nervous tissues. These specialized respectively and are responsible for movement and impulse conduction. Reproduction and development. Most animals reproduce sexually, and the diploid stage, sexual dominates the life cycle. In most species, a small flagellate fertilizes a larger non-motile egg, forming a diploid zygote. The zygote then undergoes cleavage, a succession of mitotic cell divisions without cell growth between division cell cycles. During the development of most animals, cleavage leads to the formation of multicellular stage called a blastule or blastula, which in many animals takes the form of hollow ball. Following the blastule stage is the process of gastrulation or gastrulation, during which layers of embryonic tissues that will develop into adult body parts are produced. The resulting developmental stage is called gastrula. Some animals develop directly through transient stages of maturation into adults, but the life cycles of many animals also include at least one larva stage. A larva is a sexually mature form of an animal that is morphologically distinct from the adult stage, usually eats different food and may even have a different habitat than the adult, as in the case of the aquatic tadpole larva of the terrestrial frog. Animal larvae eventually undergo metamorphosis, resurgence of development that transforms the animal into an adult. Despite the extraordinary diversity of morphology exhibited by adult animals, the underlying genetic network that controls animal development has been relatively conserved. All eukaryotes have genes that regulate the expression of other genes, and many of these regulatory genes contain common moduli of DNA sequences called homoboxes. Animals share a unique homobox containing family of genes known as Hox genes, suggesting that this gene family evolved in the eukaryotes lineage that gave rise to animals. Hox genes play important roles in the development of animal embryos, controlling the expression of dozens or even hundreds of other genes. Hox genes can thus control cell division and differentiation, producing different morphological features of animals. The sponges, which represent the lineage of simplest extant living animals, have Hox genes that regulate the formation of water channels in the body wall, the primary feature of sponge morphology. In more complex animals, the Hox gene family underwent further duplications, yielding a more versatile toolkit for regulating development. In bilaterians, a grouping that includes vertebrates, insects, and most other animals, Hox genes regulate patterning of the anterior posterior axis, as well as other aspects of development. The same conserved genetic network governs the development of both a fly and a human, despite their obvious differences and hundreds of millions of years of divergent evolution. Notice this the early embryonic development in animals. Notice also the development. Most animals also undergo gastrulation, rearrangement of the embryo, in which one end of the embryo folds inward, expands, and eventually fills the blastocoel, producing layers of embryonic tissues, the ectoderm outer layer and the endoderm inner layer. Okay, thanks.